In this video, we will learn about ABO blood groups. In humans, we know we have mainly four types of blood groups: A, B, AB, and O. And this type of blood grouping is known as ABO blood grouping. And this was discovered by Karl Landsteiner in 1900. Now, these blood groups are determined by glycoproteins present on the surface of red blood cells, and they are known as antigens or agglutinogens. Now, gamma globulin proteins in the blood plasma are known as antibodies or agglutinins. So, these antibodies found in plasma will attach to specific antigens. Now, depending on which glycoprotein is present on the red blood cell, the four blood groups are determined in humans. So, these four blood groups are determined by two types of antigen present on RBC. One is antigen A and other is antigen B. So, two types of antigens, that is A and B, and two types of antibodies, that is antibody A and antibody B, are found in human blood. Now, if we see how they are grouped into AB and O blood group. So, based on presence or absence of this antigen, human blood group falls into four different categories. So, first is a person who has antigen A belongs to blood group A. That means on the RBC cells, you will find antigen A, the protein, glycoprotein type A. So, that is blood group A. Now, if we see a person having a RBC containing B antigen, so their blood group will be called as B. Next, a person who has both antigen A and B will belong to blood group AB. That means on the same RBC, you will find A as well as B antigen. And person with no antigen on the RBC is O blood groups. O refers to 0. There is neither A antigen nor B. So this forms ABO system of blood grouping. Now if we see antigen and antibody interaction, I said antibodies are specific to specific antigen. A person with one antigen have natural antibodies in their plasma against other antigen. That means a person with antigen A will have antibodies for antigen B. So we can tell the RBC has antigen A on it, whereas the blood plasma will have antibodies for antigen B. Now the person who has antigen B, that is blood group B person, will have the antibodies for antigen A. So they have antibody A or anti-A in their plasma. Now if I take a person who has both antigen A and antigen B, so in that case the blood plasma will not have any of the two antibodies. On the contrary, person with no antigen, that is O blood group person, can have both type of antibodies, that is anti-A and anti-B. This is because antibody B, if I take, so it will go and bind to antigen B and it can form agglutination or clumping. So what is clumping reaction? So if we see antigen A, that is a person having blood group A, and antibody A, that is person having blood group B, are said to be incompatible. So similarly, antigen B and antibody B are also incompatible. That means if I have antigen A and if I have a antibody A, then what is the probability? This antibody will recognize the antigen and it will bind to it and causes clumping or agglutination. So basically similar to clotting. Similarly, a B antigen can get recognized by a B antibody and can cause clumping. So B blood group person cannot donate blood to A blood group person because B blood group person will have antibody A. So when blood from two different blood groups, that is A and B, are mixed, clumping occurs due to antigen-antibody interaction, and this is known as clumping reaction or agglutination. I hope it is clear to you. Thank you.